You feel it coming, that scratch in your throat, the exhaustion that hits like a wave. Your nose starts running, and you know what's about to happen, a cold. But here's what most people don't understand. When that rhinovirus particle lands in your nose, your body doesn't just sit there and take it. It launches a war. 7 specific mechanisms activate simultaneously, each one more brutal than the last. These aren't gentle immune responses. These are violent, coordinated attacks designed to destroy the invader at any cost. Even if that cost is you feeling absolutely miserable for a week, let's talk about what's really happening inside your body when you catch a common cold. The truth is far more intense than anyone tells you. If you value documented immunology and respiratory infection history like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. Also, this script is researched and factual, using sources from peer-reviewed journals including PNAS published January 2015 on temperature-dependent innate defense, Molecular and Cellular Pediatrics from June 2016 on rhinovirus infection mechanisms, and PMC's comprehensive review on neutrophil extracellular traps from September 2024. These aren't popular science articles. These are primary research papers from institutions studying the exact cellular mechanisms we're discussing. Now back to the information. Number 1, interferon storms at the cellular level. The moment rhinovirus punctures through your nasal epithelial cells, those cells start screaming. Not metaphorically. They release type 1 and type 3 interferons within minutes. You have around 150 rhinovirus serotypes circulating in human populations right now. More than half of all upper respiratory infections come from these viruses. They bind to receptors on your nasal cells, ICAM1 for major group rhinoviruses, low-density lipoprotein receptor family members for minor groups, a third receptor called CDHR3 for certain strains. The virus gets inside through receptor-mediated endocytosis. Different serotypes take different routes once they're in your cells. Some release their RNA genome in early endosomes. Others wait until late endosomes. This matters because the location of genome release determines which immune pathways activate first. When that viral RNA hits the cytoplasm, your cells detect it through pattern recognition receptors, RIG-I-like receptors, melanoma differentiation associated protein 5. These receptors have specific binding domains. RIG-I has a caspase activation and recruitment domain, CARD. 2 CARD domains, actually. These domains interact with MAVS, mitochondrial antiviral signaling protein. MAVS sits on the outer mitochondrial membrane. When RIG-I binds viral RNA, it oligomerizes, forms long filaments. These filaments recruit MAVS. MAVS then forms prion-like aggregates. These aggregates activate downstream signaling. Studies from 2011 show this prion-like activation mechanism. It's incredibly efficient. One RIG-I activation event can trigger massive MAVS aggregation. This amplifies the signal exponentially. MAVS activates multiple transcription factors. Interferon, regulatory factors 7 and 3. Nuclear factor kappa B. These transcription factors rush to the nucleus and turn on interferon genes. But rhinovirus fights back. Like other picornaviruses, it encodes proteases. These viral proteases cleave MAVS. A study from 2014 demonstrated that rhinovirus protease 3C can cut MAVS at specific sites. This disrupts the signaling cascade, reduces interferon production. The virus buys time. Your cells and the virus engage in molecular warfare. The cell tries to make interferon. The virus tries to block it. The outcome determines infection severity. Type 1 interferons include interferon alpha and interferon beta. Type 3 includes interferon lambda. These interferons don't just work in the infected cell, they get secreted. They bind to receptors on neighboring cells. Those cells activate the JAK-STAT signaling pathway, Janus kinase and signal transducer and activator of transcription proteins. This pathway turns on over 300 interferon-stimulated genes, 300 different antiviral proteins, PKR, protein kinase R. This enzyme detects double-stranded RNA and shuts down protein synthesis. The cell can't make its own proteins, but the virus can't make viral proteins either. 2'5' oligoadenylate synthetase. It makes molecules that activate RNase L. This enzyme degrades all RNA in the cell, viral and cellular. It's scorched earth. MX proteins. These GTPases directly block viral replication. They prevent viral genome transcription. Here's the thing though, temperature matters more than anyone realized. A 2015 study in PNAS showed that mouse airway cells infected with rhinovirus at 33 degrees Celsius versus 37 degrees Celsius had dramatically different responses. At body temperature, 37 degrees, The cells produced massively higher levels of interferons and interferon-stimulated genes. The cooler temperature of your nasal cavity, around 33 to 35 degrees, means a weaker interferon response. That's why rhinovirus replicates better in your nose than in your lungs. Your nose is cooler. The interferon response is suppressed. The virus thrives. A 2017 study found that exposing cells to low temperatures, around 25 degrees, impaired the interferon response even more. They expressed lower levels of myxovirus resistance protein 1 and 2, prime 5' oligoadenylate synthetase 1 after viral infection. When they added exogenous interferon beta, it reduced viral replication at 37 degrees, but not at 25 degrees. Cold temperature literally breaks your antiviral defense. But even with this temperature handicap, your interferon response still activates. And when it does, it turns infected cells into fortresses, cells that will die rather than let the virus spread. Because interferon also triggers programmed cell death pathways, apoptosis. The cell self-destructs, taking the virus with it. A 2016 study showed that at warmer temperatures, cells die faster when infected. This temperature-dependent cell death, combined with RNase L activity, restricts viral replication even without the full interferon response. Your cells are literally killing themselves to save you. Number 2, neutrophil extracellular traps that capture and kill. Within hours of infection, neutrophils flood your nasal passages. Neutrophils make up 50 to 79% of your white blood cells. They're the first responders. When they arrive at the infection site, they don't just eat the virus. They do something far more dramatic. They commit suicide in a very specific way. It's called netosis. Neutrophil extracellular trap formation. Discovered in 2004 by Brinkman and colleagues, nets are web-like structures made of DNA. The molecular mechanism involves specific enzymes. NADPH oxidase produces reactive oxygen species. These ROS are critical for classical suicidal netosis. Mice lacking NADPH oxidase cannot form nets properly. The ROS trigger a cascade. Neutrophil elastase translocates from granules to the nucleus. It degrades histones. Myeloperoxidase also enters the nucleus. Another key enzyme is peptidylarginine deaminase 4, PAD4. This enzyme converts arginine residues in histones to citrulline. Citrulline lacks the positive charge of arginine. This reduces the electrostatic interaction between histones and DNA. The chromatin decondenses, becomes less compact. Studies from 2018 showed that PAD4 knockout mice have impaired net formation. The chromatin doesn't decondense properly. The DNA mixes with antimicrobial proteins from the neutrophil's granules, neutrophil elastase, cathepsin G, myeloperoxidase, lactoferrin, gelatinase, cathepsin D. These proteins have high affinity for DNA. They bind to the chromatin as it decondenses. The nuclear envelope breaks down. The distinction between nucleus and cytoplasm disappears. The cell membrane ruptures. The DNA protein complex explodes outward into the extracellular space. It forms fibers, 15 to 17 nanometers in diameter. These fibers spread out like a spider web. They physically trap pathogens, bacteria, fungi, viruses. The antimicrobial proteins coating the DNA kill whatever gets caught. Nets can capture HIV virions and destroy them. They trap influenza particles. They immobilize rhinovirus. During a cold, neutrophil levels in your nasal secretions skyrocket. Sputum samples from rhinovirus-infected patients show remarkable elevation of total cells, leukocytes, and specifically neutrophils. Those neutrophils are making nets. There are actually 3 types of netosis. Suicidal netosis takes several hours. The neutrophil dies in the process. The nucleus breaks down completely. Vital netosis happens in minutes. The neutrophil survives. It packages chromatin with granule proteins into vesicles. These vesicles bud off from the nucleus. They get expelled from the cell. The neutrophil keeps functioning. It can continue immune surveillance. Mitochondrial netosis uses mitochondrial DNA instead of nuclear DNA. This creates extracellular mitochondrial DNA in the plasma. In sepsis patients, levels correlate with disease severity. All 3 types happen during respiratory infections. Studies using rhinovirus 16 challenge in asthma patients showed higher levels of lavage double-stranded DNA. This DNA came from nets. It correlated with cold symptom severity. It correlated with Th2 cytokine presence. It correlated with asthma exacerbation. When researchers treated mice with DNase to remove nets, or elastase inhibitors to prevent net formation, the Th2 immune response decreased. Cytokine production dropped. Cell recruitment diminished. But here's the problem. Nets have a dark side. The DNA backbone can chelate divalent metal cations. High concentrations destroy bacterial membranes, But extracellular DNA also triggers inflammatory responses. If it's not cleared properly by circulating nucleuses and phagocytes, it amplifies inflammation. Mitochondrial DNA acts as a damage-associated molecular pattern. It stimulates more neutrophils to release more nets through positive feedback. This creates a vicious cycle. Nets also contain histones. Histones are strongly cationic. They're directly toxic to cell membranes. During infections, netosis becomes the predominant source of extracellular histones, over 80%. These histones promote endothelial dysfunction. They activate platelets. They activate more neutrophils. They bind to toll-like receptors 2 and 4 on other immune cells. This activates the mitogen-activated protein kinase pathway. It activates the NLRP3 inflammasome. More inflammation, more cytokines, more tissue damage. Nets can even trap your own immune cells. CD4 positive and CD8 positive T cells, B cells, monocytes. During HIV infection, this contributes to immune cell depletion. The sticky, charged nature of nets creates problems in cystic fibrosis. They increase sputum viscosity. They obstruct airways. In your nose during a cold, nets capture rhinovirus particles. They prevent viral spread. They kill infected cells. But they also contribute to the inflammation that makes you feel terrible. The congestion, the pain, the tissue damage. Your body accepts this collateral damage. It's worth it to stop the virus. Number 3, fever mechanisms that cook the virus. You feel cold even though your body temperature is climbing. That's because your hypothalamus just reset its thermostat. The preoptic area of your anterior hypothalamus controls body temperature. It has thermosensitive neurons. During infection, these neurons receive signals from the periphery. Immune cells activated by viral proteins release cytokines, interleukin-1 beta, interleukin-6, tumor necrosis factor, alpha. These are endogenous pyrogens, fever-producing molecules made by your own cells. Macrophages produce them first. Then T lymphocytes, B lymphocytes, other leukocytes join in. At least 11 different cytokines can initiate fever, and these cytokines travel through your bloodstream to your brain. But there's a problem. They can't cross the blood-brain barrier. Most of them are too large. So how do they signal the brain to induce fever? Multiple pathways. First, cytokines can bind to receptors on endothelial cells lining brain blood vessels. This stimulates those endothelial cells to produce prostaglandin E2, PGE2. This prostaglandin crosses into the brain parenchyma. It reaches the preoptic hypothalamus. Second, cytokines can activate neurons in circumventricular organs. These are brain regions with fenestrated capillaries, leaky blood vessels. The organum vasculosum of the lamina terminalis is one. It lacks a normal blood-brain barrier. Cytokines access it directly. These neurons then signal deeper brain structures. Third, cytokines can activate peripheral nerves, vagal afferents. These nerves carry the immune signal to the central nervous system. Fourth, some cytokines do cross the blood-brain barrier through saturable transport mechanisms. Interleukin-1 alpha, interleukin-1 beta, they get actively transported across. Once in the brain, these signals trigger prostaglandin E2 synthesis. The enzyme cyclooxygenase 2, COX2, converts arachidonic acid to prostaglandin H2. Microsomal prostaglandin E synthase 1 converts this to PGE2. But let's break down this pathway further. Arachidonic acid comes from membrane phospholipids. Phospholipase A2 releases it. This happens when cytokines bind to endothelial cell receptors. The arachidonic acid pathway has multiple branches. COX1 and QX2 can both convert arachidonic acid. COX1 is constitutively expressed, always present. COX2 is inducible, gets upregulated during inflammation. In brain endothelial cells during infection, QX2 expression increases dramatically. Studies using immunohistochemistry show intense COX2 staining in blood vessel walls after lipopolysaccharide injection. The enzyme produces prostaglandin H2. This intermediate is unstable. Multiple synthases can convert it. Prostaglandin E synthase makes PGE2. Prostaglandin D synthase makes PGD2. Prostaglandin I synthase makes prostacyclin. Thromboxane synthase makes thromboxane A2. During fever, the PGE2 pathway dominates. Studies from 2015 show that prostaglandin E2 binding in the preoptic hypothalamus produces fever by disinhibition of presympathetic neurons in the brainstem. These neurons normally inhibit thermogenesis. PGE2 removes that inhibition. Heat production activates. The prostaglandin binds to EP3 receptors. Prostaglandin E2 receptor subtype 3. These receptors are concentrated in the preoptic hypothalamus. When activated, they raise the thermoregulatory set point. Your brain now thinks normal body temperature is too cold. It activates heat production mechanisms, shivering thermogenesis. Your muscles contract rapidly. This generates heat. Brown adipose tissue activation. This tissue burns calories purely to make heat. Vasoconstriction. Blood vessels in your skin constrict. This reduces heat loss. Your core temperature climbs from 37 degrees Celsius to 38 or 39 degrees, sometimes 40 degrees in severe infections. Studies using experimental rhinovirus infection show fever patterns. Temperature rises within hours of viral inoculation. It peaks around day 3. The timing matches the cytokine response. Blood samples show elevated interleukin 8, 7-fold increase on day 3. Tumor necrosis factor increases. Interleukin 6 increases. These cytokine levels correlate with fever magnitude. Ketoprofen, a prostaglandin synthesis inhibitor, abolishes fever. It blocks the temperature rise to lipopolysaccharide, to polyinosinic polycytidylic acid, to interleukin 1, to tumor necrosis factor alpha, block prostaglandin production, no fever. But interestingly, COX inhibitors don't completely eliminate interleukin 1 beta-induced fever in some studies. A 2022 study showed that indomethacin pretreatment didn't prevent brown adipose tissue thermogenesis or cutaneous vasoconstriction in response to intravenous interleukin 1 beta. This suggests COX-independent fever mechanisms exist. Interleukin 1 beta can activate neurons in the dorsomedial hypothalamus. These neurons project to the rostral rapha pallidus area. These areas control sympathetic outflow to brown adipose tissue and cutaneous vasculature. Glutamate receptor activation in the dorsomedial hypothalamus is required for this COX-independent fever. So your fever works through multiple redundant pathways. Block one, others compensate. Why does your body do this? Why waste energy raising temperature? Because it works. Rhinovirus replication is temperature sensitive. It grows better at 33 to 35 degrees, worse at 37 degrees. Raise your body temperature to 38 or 39 degrees, viral replication drops further. The interferon response strengthens at higher temperatures. Immune cell function improves. Neutrophil activity increases. Lymphocyte proliferation accelerates. Antibody production goes up. Fever is a weapon. It makes your body inhospitable to the virus. The virus evolved to thrive in your cool nasal passages. Your body responds by cooking those passages. You feel miserable because that's the point. The discomfort keeps you resting, conserving energy, letting your immune system work. Number 4, mucus hypersecretion that drowns invaders. Your nose becomes a faucet. Clear fluid pours out constantly. Then it turns thick, yellow, green. This isn't random. This is a highly regulated defense mechanism. Goblet cells in your nasal epithelium normally produce baseline mucus. Submucosal glands contribute too. This mucus forms a protective layer. It traps particles, dust, bacteria, allergens. Cilia on epithelial cells beat in coordinated waves. Mucociliary clearance, they move the mucus toward your throat. You swallow it. This happens constantly. You don't notice. During rhinovirus infection, everything changes. Viral replication in ciliated epithelial cells triggers signaling pathways. Inflammatory mediators get released. Interleukin 1 beta, tumor necrosis factor, interleukin 8, interleukin 6, interleukin 11, chemokines, rantas, monocyte chemoattractant protein 1, macrophage inflammatory protein 10, vasoactive peptides, bradykinin, growth factors, vascular endothelial growth factor. These mediators activate inflammatory cells. Leukocytes invade the submucosa, granulocytes, monocytes. This amplifies the inflammatory process. Neutrophils flood in. They release more inflammatory signals. The toll-like receptor pathways activate. TLR3 recognizes viral double-stranded RNA. This triggers transcription factor activation. Nuclear factor kappa B. This upregulates mucin gene expression, but the regulation is complex. The MUC5AC gene promoter contains multiple binding sites, NF-kappa B binding sites, activator protein 1, AP1 sites, STAT6 binding sites. Different stimuli activate different transcription factors. These bind to the promoter. Increase gene transcription. Studies using chromatin immunoprecipitation showed that during viral infection, NF-kappa B binds strongly to the MUC5AC promoter. Histone acetylation increases at this locus. The chromatin opens up. Transcription machinery accesses the gene more easily. Messenger RNA production skyrockets. The mRNA gets translated. MUC5AC protein is massive, hundreds of thousands of Daltons. It has a protein backbone with heavily glycosylated regions. Oligosaccharide chains cover it. These chains are negatively charged. They bind water. This creates the gel-like properties of mucus. Not just production increases, goblet cell numbers increase too. Goblet cell hyperplasia. Normally, goblet cells are scattered among ciliated epithelial cells. During chronic inflammation or repeated infections, more epithelial cells differentiate into goblet cells. The ratio shifts. Studies in chronic obstructive pulmonary disease show this clearly. Airway biopsies reveal increased goblet cell density. Same happens with repeated rhinovirus infections. MUC5AC is the major mucin in airway secretions. During rhinovirus infection, MUC5AC gene levels skyrocket. Also MUC5B, also GOB5. These genes encode the protein backbone of mucus. Studies show that patients with rhinovirus infection have remarkable elevation of mucus-related genes. Bronchiolar samples from infected infants show increased mucus secretion. Cell debris, mechanical airway obstruction results. The epidermal growth factor receptor pathway contributes. EGFR. Rhinovirus activates this pathway. EGFR inhibition suppresses virus-induced mucin expression in mice. The JAK-STAT pathway is involved. Janus kinase and signal transducer and activator of transcription proteins. Th2 and Th17 cytokines activate these. Interleukin-5, interleukin-13, interleukin-17. These drive mucus hypersecretion. The NLRP3 inflammasome activates. This protein complex responds to viral components. It produces interleukin-1 beta, interleukin-18. These cytokines further stimulate mucin production. Studies using nasal lavage fluid from experimentally infected subjects show changes over time. Day 0, minimal mucus. Day 3, maximum changes. Gel phase mucin increases twofold. Total protein doubles. Immunoglobulin, G, a marker of vascular leak, increases fourfold. Interleukin-8 increases sevenfold. Lysozyme from Ceramicus cells increases. Alcin blue staining of acidic mucoglycoproteins intensifies. This corresponds to symptom progression. Early infection produces watery rhinorrhea, thin, clear fluid. This is mostly plasma exudate. Increased vascular permeability lets fluid leak from blood vessels. As infection progresses, mucin secretion dominates. The discharge becomes thick, mucoid. Neutrophils infiltrate. The mucus turns yellow, then green. Neutrophil enzymes, myeloperoxidase. The color comes from cellular debris. Cold temperatures enhance mucus production through additional mechanisms. A protein called cold inducible RNA binding protein, SIRP, gets overexpressed. Cold stress causes SIRP to translocate from the nucleus to the cytoplasm. This depends on arginine methylation. In the cytoplasm, CARP stabilizes pro-inflammatory cytokine messenger RNAs. It binds to 3' untranslated regions of these mRNAs. This increases their translation. More cytokines, more MUC5AC production. A 2016 study showed that CARP gene and protein levels were significantly increased in COPD patients and in rats with chronic airway inflammation. Cigarette smoke inhalation upregulated inflammatory cytokine production and MUC5AC secretion. Cold temperature amplified this. CARP silencing or treatment with a methyltransferase inhibitor promoted cytokine mRNA degradation. This inhibited inflammation and mucus hypersecretion. Another mechanism involves transient receptor potential melastatin-8, TRPM8. This calcium permeable cation channel is activated by cold temperatures. It's expressed in bronchial epithelium. It's upregulated in COPD subjects. Exposure to cold temperatures induces ciliary ultrastructural anomalies. Mucus accumulates on the epithelial surface. Cold air provokes airway mucus hypersecretion through TRPM8 mediated calcium influx. Why does your body do this? Mucus traps viral particles. It contains antimicrobial proteins, lysozyme, lactoferrin, immunoglobulins. These proteins neutralize viruses. The thick mucus physically blocks viral access to epithelial cells. Mucociliary clearance moves virus-laden mucus out of your airways. You cough it up, you blow it out, you swallow it, stomach acid destroys it. The downside is obvious. You can't breathe through your nose. You can't taste food. You feel congested. Pressure builds. Your head hurts. But this mechanism works. It physically removes millions of viral particles every hour. It's mechanical warfare. Drown the enemy in mucus. If you value this documented cellular biology like this, subscribe. It's the best way to support our work. These mechanisms we're discussing aren't theory. They're documented in studies from clinical laboratories around the world. The mucus data comes from a 2021 Frontiers in Immunology systematic review on abnormal airway mucus secretion induced by viral infection. The neutrophil research comes from a Nature Signal Transduction and Targeted Therapy review from September 2024. These are the sources you won't find in popular articles. Now back to the mechanisms your body uses to destroy this virus. Number 5, antibody production that marks for destruction. Around day 3 of infection, your adaptive immune system joins the fight. B lymphocytes in your nasal mucosa detect viral antigens. These B cells have surface receptors, B cell receptors. Each B cell has a unique receptor recognizing a specific antigen. When a B cell receptor binds its matching viral antigen, that B cell activates. It receives help from T helper cells, CD4 positive cells. These T cells recognize viral peptides presented on MHC class II molecules. The interaction provides signals, cytokines, CD40 ligand. The B cell proliferates. It makes copies of itself. These clones differentiate. Some become plasma cells, antibody factories. Some become memory B cells, long-lived cells that remember this virus, but the process of class switching deserves explanation. Initially, all B cells make IgM. The immunoglobulin heavy chain gene has multiple constant region segments, mu, delta, gamma, epsilon, alpha. These encode different antibody classes. Through a process called class switch recombination, the B cell can change which constant region it uses. It cuts out DNA between the current constant region and a downstream one, permanently deletes that DNA. Rejoins the remaining segments. This is irreversible. The enzyme activation-induced cytidine deaminase, AIDE, mediates this process. It creates DNA breaks at specific switch regions. Studies from 2017 showed that cytokine signals determine which class the B cell switches to, transforming growth factor beta, plus interleukin 5 or interleukin 10 promotes IgA switching. Important for mucosal immunity. Interleukin 4 promotes IgE switching, relevant for allergic responses. Interferon gamma promotes IgG switching, particularly IgG1 and IgG3 subclasses. During rhinovirus infection, the mucosal environment provides signals for IgA switching. This makes sense. IgA is the primary mucosal antibody. Plasma cells produce immunoglobulins. Antibodies. 5 main classes exist: IgM, IgD, IgE, IgG, IgA. Each has different functions. IgM appears first. It's the initial antibody during a primary response. B cells always make IgM first before class switching. IgM is huge. It's a pentamer. 5 Y-shaped units joined together, 10 antigen binding sites. This massive size makes it excellent at binding bacterial surfaces. It's great at activating complement. It agglutinates pathogens, clumps them together. Studies show that after rhinovirus inoculation, B cell responses can be detected by day 3. Mucosal IgA production starts, then IgM appears. Finally, IgG by day 7 to 8. IgA is critical for respiratory immunity. It's the principal antibody in secretions: saliva, tears, respiratory secretions, intestinal secretions. In blood, IgA is monomeric, single Y-shaped molecule. In secretions, it's dimeric, 2 IgA molecules joined by a J-chain protein. Secretory component protects it from degradation. This dimeric secretory, IgA, Zyga, is produced by plasma cells in the lamina propria of mucosal membranes. The IgA dimer binds to polymeric immunoglobulin receptor on the basolateral side of epithelial cells. The receptor transports the IgA across the epithelial cell barrier. Gets released on the mucosal surface. More IgA is synthesized daily than any other antibody class, but it's not as stable as IgG, so levels stay relatively low at any given time. Secretory IgA prevents viral particles from adhering to mucosal surfaces, neutralizes them, facilitates their removal by mucociliary clearance. Specific IgA to rhinovirus appears in nasal secretions within days. Elevated serum titers of serotype-specific IgA correlate with reduced viral shedding. Studies show that nasal epithelial cells express the polymeric immunoglobulin receptor at their ciliated side. All cells can transport IgA to the mucosal surface. IgG production follows. IgG is the main blood antibody. 4 subclasses exist: IgG1, IgG2, IgG3, IgG4. IgG1 is about 65% of total IgG. IgG1 and IgG3 activate complement powerfully. They bind Fc receptors on immune cells strongly. They're central in clearing viral and bacterial infections. IgG2 specializes in recognizing polysaccharide antigens, important for encapsulated bacteria. IgG4 is less inflammatory, associated with tolerance. Rapid induction of specific preformed neutralizing IgG to rhinovirus prevents or limits reinfection. Elevated serum titers of serotype-specific IgG neutralizing antibody correlate with attenuated cold symptoms. Reduced viral shedding. The antibodies work multiple ways. Neutralization. The antibody binds to viral surface proteins, blocks attachment sites. The virus can't bind to cell receptors, can't enter cells, can't replicate. Opsonization. Antibodies coat viral particles. The Fc region of IgG binds to Fc receptors on phagocytes, macrophages, neutrophils. These cells recognize the antibody-coated virus, engulf it, destroy it. This is antibody-dependent cellular phagocytosis, complement activation. IgM and IgG activate the complement cascade, a series of protein reactions. Complement proteins coat the pathogen, form the membrane attack complex. This punches holes in viral envelopes, destroys the virus. Complement fragments also act as opsonins, enhance phagocytosis. The problem with rhinovirus is antigenic diversity. Around 160 serotypes exist. Each has slightly different surface proteins. Antibodies to one serotype don't protect against others. You can get infected repeatedly, different serotypes. That's why there's no rhinovirus vaccine. Too many targets. But within a serotype, immunity is strong. If you get infected with serotype 16, you develop antibodies. Those antibodies protect against serotype 16 for years, maybe life. The issue is you'll encounter serotype 14 next time, or serotype 39, different virus, no cross-protection. Memory B cells remain in lymphoid tissues, in mucosal sites. If you encounter the same serotype again, those memory cells activate rapidly. They differentiate into plasma cells faster. They make IgG or IgA immediately. Skip the IgM phase. Secondary immune response, much quicker, much stronger. The virus gets neutralized before symptoms develop. This is why repeated infections with the exact same rhinovirus serotype are rare. Your antibodies, remember, they're ready. They destroy the virus on contact. But with 160 serotypes circulating, you're always vulnerable to a new one. Number 6, vascular permeability changes that weaponize inflammation. Your nose feels stuffed. Your face throbs. Your head pounds. This isn't just congestion. This is inflammation. Specifically, increased vascular permeability. Your blood vessels are leaking. Normally, endothelial cells lining blood vessels form tight barriers. Adherens junctions connect the cells. VE cadarin proteins link adjacent cells. These junctions control what passes from blood into tissues. But it's not just VE cadarin. Multiple junction proteins exist. Tight junction proteins include claudins, occludin, and junctional adhesion molecules. These create the seal between cells. Adherens junctions include VE cadarin, which connects to the actin cytoskeleton through catenins, alpha-catenin, beta-catenin, 120-catenin. These form a complex. The complex anchors to actin filaments inside the cell. This creates mechanical strength. During infection, inflammatory mediators disrupt these junctions. The signaling pathways are complex. Histamine binding to H1 receptor activates phospholipase C. This cleaves phosphatidylinositol 4,5-bisphosphate into inositol triphosphate and diacylglycerol. IP3 triggers calcium release from endoplasmic reticulum. Calcium activates myosin light chain kinase. This enzyme phosphorylates myosin light chains. Phosphorylated myosin interacts with actin. The cell contracts. Simultaneously, diacylglycerol activates protein kinase C. PKC phosphorylates multiple targets, including VE cadarin. Phosphorylation promotes VE cadarin internalization. The protein gets pulled away from cell junctions. Gaps form between cells. Studies using transendothelial electrical resistance measurements confirm this. Histamine treatment decreases resistance, indicates barrier breakdown. Histamine is the first actor. Mast cells in nasal tissue detect injury signals. They degranulate, release histamine. Histamine is a biogenic vasoactive amine. It causes vasodilation. It increases vascular permeability. It binds to histamine receptors. H1 receptors on venular endothelial cells, H2 receptors in some tissues. H1 receptor is a G protein coupled receptor, uses GACU 11 family proteins. Activation promotes calcium flux. This triggers endothelial cell contraction. Gaps form between cells. Studies using intravital imaging show that histamine dilates vasculature, increases blood flow, induces hyperpermeability specifically in venules, not arterioles. Whole mount immunostaining shows VE cadarin localization changes. Normally, VE cadarin sits at cell-cell contact areas. After histamine exposure, partial internalization occurs. VE cadarin disassembles. The endothelial barrier breaks down, fluid leaks out, plasma proteins leak out. This happens within 10 minutes. Histamine has a half-life of about 1 minute. Effects are rapid. Effects also resolve quickly. This is critical for vascular homeostasis. Can't have permanent leakage. The mechanism involves multiple pathways: nitric oxide-dependent vascular dilation, increased blood flow, PKC, protein kinase, CAO, and ROC, Rho-associated protein kinase, ROS pathways. These disrupt endothelial barriers. Inhibiting nitric oxide synthesis with L-name significantly reduces histamine-induced hyperpermeability, but doesn't completely abolish it. Vasoconstriction with phenylephrine also reduces leakage, but endothelial barrier disruption still occurs. Both mechanisms contribute. Increased flow, disrupted barriers. Bradykinin amplifies this. Bradykinin is generated in tissues after mast cell degranulation. It's a potent vasoactive substance, induces burning pain, increases vascular permeability by disrupting endothelial barriers. Similar to histamine, studies show bradykinin causes VE cadherin disassembly at cell-cell junctions. Prostaglandins modulate permeability through altering blood flow, prostaglandin E type especially. When injected alone, they have little effect on permeability. But they significantly augment the permeability induced by histamine or bradykinin. Prostaglandins are more potent at inducing hyperemia than histamine or bradykinin. Hyperemia means increased blood flow. There's a correlation between hyperemia degree and the augmentation of enhanced vascular permeability. More blood flow, more leakage. During rhinovirus infection, all these mediators flood your nasal tissue. Infected epithelial cells release cytokines. Interleukin-1 beta, tumor necrosis factor. Interleukin-8, interleukin-6. These activate immune cells. Immune cells release histamine, bradykinin, prostaglandins, leukotrienes, C4 and D4. These also increase permeability, platelet activating factor, released during cold urticaria. Increases permeability. The result is tissue edema. Swelling? Nasal lavage fluid analysis shows the damage. Total protein increases 2-fold. IgG increases 4-fold. IgG in nasal secretions means vascular leak. IgG doesn't belong there. It's a blood protein. Its presence proves barriers broke down. This vascular leak serves purposes: dilutes toxins, dilutes bacterial products, brings complement proteins to infection site, brings antibodies, brings more immune cells. Neutrophils migrate from blood into tissue through these gaps. They follow chemokine gradients. Interleukin-8 is a powerful neutrophil chemoattractant. Experimental rhinovirus infection shows IL-8 increases 7-fold by day 3. That's a huge signal. Neutrophils pour through leaky vessel walls, enter infected tissue, start killing virus. The downside is the edema, swollen tissue, blocked nasal passages. You can't breathe. Blood vessels in your face leak too. Facial pressure, sinus pressure, headaches. The inflammation causes pain, activates nerve pain receptors. Bradykinin especially causes burning pain. Prostaglandins sensitize pain receptors. Everything hurts. Your body accepts this. The temporary discomfort is worth stopping the infection. Leak the vessels, flood the tissue with immune cells, destroy the virus. Collateral damage to your comfort is acceptable. Number 7. Cytokine cascades that orchestrate the war. Every mechanism we've discussed is controlled by cytokines. These small proteins are intercellular communication molecules. They coordinate immune responses. During rhinovirus infection, cytokine production explodes. Type 1 interferons. Interferon alpha, interferon beta, produced within hours. Type 3 interferons, interferon lambda. Also early, these trigger interferon-stimulated gene expression, over 300 genes, antiviral proteins. They also signal neighboring cells. Create an antiviral state throughout the tissue. Pro-inflammatory cytokines follow. Interleukin-1 beta, one of the most important endogenous pyrogens, stimulates T lymphocyte proliferation in presence of antigen, enhances immune response, initiates fever through prostaglandin E2 synthesis in the hypothalamus, activates vascular endothelial cells, increases adhesion molecule expression, promotes neutrophil recruitment, tumor necrosis factor alpha, another major endogenous pyrogen, reproduces many physiologic derangements of septic shock, mediates deleterious effects of gram-negative infections, activates endothelial cells, increases vascular permeability, promotes inflammation, interleukin-6, key pyrogenic cytokine. Serum levels correlate with fever magnitude. Activates acute phase protein production in liver, promotes B-cell differentiation, enhances antibody production, interleukin-8, powerful neutrophil chemoattractant, 7-fold increase on day 3 of rhinovirus infection, creates gradient, pulling neutrophils to infection site, also activates neutrophils, enhances killing capacity, chemokines, direct cell traffic, orantes, regulated on activation, normal T cell expressed and secreted, attracts T cells, eosinophils, monocytes, MCP1, monocyte chemoattractant protein 1, brings monocytes to tissue. These differentiate into macrophages. MIP10, macrophage inflammatory protein, recruits more immune cells, vasoactive peptides. Bradykinin increases vascular permeability, causes pain, contributes to edema, growth factors. Vascular endothelial growth factor promotes angiogenesis, increases vascular permeability, helps deliver immune cells to infected tissue. Studies analyzing these mediators show patterns. Immediate early cytokines, interferons. These appear within hours, peak early, start declining by day 2. Early cytokines, IL-1 beta, TNF alpha, IL-6. These rise rapidly, peak day 2 to 3. Sustained cytokines, IL-8, chemokines. These maintain elevation through day 5 to 7, keep recruiting immune cells. Late cytokines, interleukin-10, this is anti-inflammatory, begins rising day 3, peaks day 5. Starts shutting down inflammation. IL-10 inhibits production of pro-inflammatory cytokines. IL-1, IL-6, TNF alpha. IL-10, knockout mice suffer chronic inflammatory bowel disease. In rhinovirus infection, IL-10 production by infiltrating monocytes may decrease local inflammatory reaction. Inhibit cellular immune responses. It's a negative feedback loop. Initial inflammation is beneficial, recruits immune cells, kills virus, but prolonged inflammation damages tissue. IL-10 applies brakes, prevents excessive damage. The cytokine response varies by individual. Studies comparing infected allergic subjects to non-allergic subjects found no differences in cytokine patterns, but asthma patients show altered responses. Some studies report impaired interferon production in asthmatics during rhinovirus infection. Others don't replicate this. The mechanisms behind asthma exacerbations from rhinovirus remain unclear. One theory suggests deficient innate immunity in asthmatics. TLR7 signaling impairment. TLR7 ligation induces type 1 interferon production. Mice with impaired TLR7 signaling show reduced interferon production after rhinovirus. Also show eosinophilia, airway hyperreactivity. Effects reverse with adoptive transfer of TLR7 competent plasmacytoid dendritic cells, or exogenous interferon. Th2 cytokine induction negatively regulates TLR7 or TLR3 induced interferons. IL-4 and IL-13 inhibit interferon production. Asthmatics have baseline Th2 skewing. This may impair their antiviral responses, make them more susceptible to severe rhinovirus infections. Another phenomenon is neutrophil extracellular trap-associated inflammation in asthmatics. Rhinovirus 16 challenge induces NET-associated double-stranded DNA in airways. Higher levels in asthmatics correlates with cold symptom severity, with Th2 cytokine presence. With asthma exacerbation, NETs amplify Th2 responses, create a pro-inflammatory environment. Treating with DNase or elastase inhibitors reduces this. The cytokine cascade is self-limiting in healthy individuals. Initial infection triggers strong pro-inflammatory response. This recruits immune cells, activates killing mechanisms, clears virus. By day 5 to 7 hours, viral titers drop. Cytokine production decreases. Anti-inflammatory cytokines rise. IL-10. Resolution phase begins. Tissue repair starts. Symptoms resolve. The whole process takes 7 to 10 days typically. Some symptoms linger longer. Post-infectious cough can persist weeks. Residual airway inflammation. But the acute phase ends. Your immune system wins. The virus is cleared. Your body returns to baseline. Here's what they don't tell you. These 7 mechanisms aren't gentle. They're not surgical strikes. They're carpet bombing. Your body destroys infected cells, kills bystander cells, floods tissues with inflammatory mediators, raises temperature, makes you miserable, all to kill a virus that isn't even directly cytopathic. Rhinovirus doesn't destroy your cells. Your immune response does. Studies show minimal cytopathic alterations in airway epithelium during rhinovirus infection. The absence of visible damage led to the hypothesis that symptoms come from the immune response, not the virus itself. This has been confirmed. The cold you experience, the symptoms you suffer, are almost entirely caused by your immune system fighting the virus, the interferon storms. The neutrophil traps, the fever, the mucus floods, the antibody production, the vascular leakage, the cytokine cascades, all of this causes your runny nose, your congestion, your headache, your fatigue, your sore throat, your cough. The virus just sits there replicating. Your body unleashes total war. In 2023, researchers examining the exact mechanisms found something critical. A soil core analysis of nasal tissue from chronic cold sufferers showed persistent low-level inflammation even after viral clearance. The immune response had created lasting changes. Epithelial barrier disruption, altered mucin production, enhanced sensitivity to irritants. This explains why some people suffer repeated colds more severely. Their baseline immune state never fully recovers. Each infection compounds the damage. Each immune response becomes more aggressive, more destructive. The tissue bears the scars. But here's the truth. Without these violent responses, you'd die. Before modern medicine, respiratory infections killed millions annually. They still kill vulnerable populations, infants, elderly, immunocompromised. The intensity of these 7 mechanisms is necessary. Evolution selected for aggressive immune responses because the alternative is uncontrolled viral replication, spread to lungs, pneumonia, death. Your week of misery prevents your funeral. The human body accepted this trade-off millions of years ago. We inherited these mechanisms from ancestors who survived plagues, who fought off countless pathogens, who passed these aggressive immune genes to their children. You're alive because your immune system is brutal, because it accepts collateral damage, because it values survival over comfort. The next time you feel a cold coming on, remember this. Those 7 mechanisms are already activating, already preparing for war. The interferon storm is gathering. The neutrophils are mobilizing, the temperature is rising, the mucus factories are ramping up, the antibody production lines are starting, the blood vessels are preparing to leak, the cytokine cascade is beginning. You're about to feel absolutely terrible for a week. And that's exactly what's supposed to happen. That's your body working correctly. That's billions of years of evolution unleashing coordinated destruction on a microscopic invader. That's survival. That scratchiness in your throat isn't weakness. It's the opening salvo of a war your body has fought 10,000 times before. And it's going to win this one too.